Hello, my name is Caroline Schlehuber, and today I will be telling you about my research on plants and fungi in indigenous medicine. To begin, I wanted to examine the overarching problem of the serious lack of effective antibiotics and antibacterial medicines in modern medicine. And we all are still in a pandemic, and though that pandemic is viral, it's a viral pandemic, not a bacterial one. Um, it's likely that we will have more pandemics in the future. And so this is a really big problem in our society today. The overarching solution that I kind of wanted to examine through my research was researching the medicinal practices that were already used for hundreds of years by indigenous cultures and groups. So to do this, I chose two plants and one mushroom that has history in indigenous medicine. That those are Canada goldenrod, stinging nettle, and lion's mane mushroom. And all three of these organisms have a variety of uses in indigenous medicine, such as treating burns, aiding digestion, aiding pregnancy, easing arthritis, and they were used by many different tribes throughout North America. And additionally, the lion's mane mushroom was actually used in ancient Chinese medicine to boost cognitive health as well. So the goals that I had with my experiment were first to just find some kind of bacterial resistance through inhibition assays in the specimens I chose to study. But on a larger scale, I really wanted to prove that there is so much merit and value to the medicinal practices of indigenous people, and we need to, acknowledge this in Western medicine as well. My hypothesis is that plants and mushrooms historically used in indigenous medicine will display antimicrobial properties, which therefore shows that indigenous medicine holds many promising candidates for future antimicrobial discovery. And I'll reach this goal through a series of controls, like using established scientific literature in my methods and positive and negative controls, which I will discuss in my methods. And I will know if I've had success with proof of bacterial inhibition in my specimens. Next, I started out by first just gathering the samples of these plants and mushroom in the field in a nearby park by my house. And I prepared and stored them by sterilizing and freezing and refrigerating the plants and dehydrating the fungi to make them stable over time. I then placed the plant samples on cornmeal agar, and I was able to grow endophytic fungi out of them, which means the fungi that were living inside of the plants, not on the outside, because I sterilized the plant samples. And then I took plugs of that endophytic fungi, placed them on potato dextrose agar, and isolated them from the plant. I then derived an extract of the lion's mane mushroom, Paramecium erinaceae, for testing by grinding it up, rehydrating, concentrating, and centrifuging it. And then I performed inhibition assays on both the endophytic fungi from the plants and the mushroom extract that I created against two strains of bacteria in some different ways that I'll explain later. I then measured inhibition rings and I came to conclusions. And the inhibition rings show that the fungi is able to fight off the bacteria that I was testing against. So here is how I was able to grow endophytic fungi out of the plants. The top row of images are the plant samples, and you can kind of see some cloudy white spaces surrounding them. And below are the fungi that I isolated from them by taking samples. And those cloudy white areas I mentioned are hyphal threads of endophytic fungi growing out of the plants. So next I'd like to discuss my results, first from the endophytic fungi that I grew out of the plant samples that I collected. So I did test it against some strains of bacteria, Enterobacter aerogenes and Staph epidermidis, and there wasn't really enough results or evidence to draw conclusions from that. But what I wasn't expecting to find were antifungal properties So this plate that you can see here has a lot of different samples of fungi on it, and that were extracted from the plants. And I'd like you to focus on sample A, which is encircled in red. And it's originally grown from Solidago canadensis, or the goldenrod plant I collected, and it shows strong potential for antifungal properties based on this clearing of inhibition displayed. And it continued to inhibit the growth of the other fungi for upwards of 2 to 3 months, or the duration of my experiment. And because it was such a clear ring and it's still present, it's been present for so long, this shows that this sample A, which does remain unidentified, does have strong potential for antifungal properties. And next, the results from the lion's mane mushroom, or the Herinaceum erinaceae extract. So how I did this testing was I soaked sterile paper discs in the extract that I created, and then I placed those discs on a lawn of bacteria. So it was a plate covered in bacteria with a sterile paper disc in the center of it, soaked in extract. And some of those paper discs did not have extract as a form of a control to prove or to show that it wasn't the paper discs causing the inhibition, but actually the extract. So I did find clear evidence of bacterial inhibition of Enterobacter aerogenes and Staph epidermidis, which is encircled in red. The rings were most present 48 hours after plating, but appeared kind of faded in the images as some time had passed. Inhibition rings were also stronger against Enterobacter aerogenes than Staph epidermidis. And I know that I have sufficient evidence to say that there is bacterial inhibition because I repeated the same test upwards of 5 times in my research. And here's some data I collected. I measured the inhibition rings in centimeters, and as you can see, the inhibition rings averaged around 1.5 centimeters for all the testing, and the size did decrease over time, which is kind of something I wasn't expecting to find as well. So based on this data, Herinaceum erinaceae does have the potential to inhibit the growth of both gram-positive and gram-negative bacteria, so a very wide variety of bacteria. This makes sense as to why these plants were used in indigenous medicine, because medicines need to have the ability to fight off bacteria, as this mushroom does. And I chose to use an extract as the form of the mushroom because in indigenous medicine, the mushroom was consumed as a tea. So that's the closest way I could kind of replicate that in my research. And even though the rings of inhibition are small, they're still significant because it shows that this mushroom can employ the use of secondary metabolites to fight off bacteria. And some sources of error in my research are contamination. I did have to throw out some data because of contamination. And I think the age of the extract, like over time, it became less effective against the bacteria. So some conclusions that I was able to draw from my research are that the sample of endophytic fungi grown from the goldenrod flower has the potential for antifungal properties. Additionally, the diameter of the inhibition rings are directly correlated to the age of the mushroom extract. Herinaceum erinaceae did inhibit the growth of Enterobacter aerogenes more than the growth of Staph epidermidis, and all of these findings do support my hypothesis that plants and fungi used in indigenous medicine have antimicrobial properties. Finally, in the future, I'd like to do these tests with less time in between, so time is less of a variable, and I would like to keep the extract in the fridge in a hope to preserve it so that it has the same effectiveness over time. And if I am to continue my research in the future, which I hope to do, I'd like to calculate the appropriate dosage of mushroom extract for it to be effective as a medicine. Here are my references, and thank you so much for watching my presentation.